What is the F508del CFTR gene mutation? Imagine a tiny change in your genes that can have a big impact on your health. That's what the F508del mutation does in the CFTR gene, which is the most common cause of cystic fibrosis. This mutation involves the deletion of 3 DNA building blocks, called nucleotides, in the CFTR gene. Because of this deletion, a single amino acid called phenylalanine is missing at position 508 in the CFTR protein. The CFTR gene provides instructions for making a protein that acts like a channel on the surface of cells lining the lungs, pancreas, and other organs. This channel helps regulate the flow of chloride and bicarbonate ions, which are essential for maintaining the right balance of salt and water on these surfaces. When the CFTR protein works properly, it keeps mucus thin and clear, making it easier for the body to clear out bacteria and other debris. However, the F508del mutation causes the CFTR protein to misfold. Normally, proteins fold into specific shapes to function correctly. When they don't fold right, the cell recognizes them as faulty and destroys them before they reach the cell surface. This means very little functional CFTR protein makes it to the cell membrane. The small amount that does reach the surface is unstable and doesn't work well, leading to poor chloride transport. Most people with cystic fibrosis inherit 2 copies of this mutation, one from each parent. This is why the F508del mutation is so common. It accounts for about 70% of all CF gene mutations worldwide and affects nearly 2/3 of people with cystic fibrosis. The lack of proper chloride transport causes thick mucus to build up in the lungs, leading to breathing problems, frequent infections, and damage to lung tissue. It also affects the pancreas, leading to difficulties in digestion and nutrient absorption, and causes elevated sweat chloride levels, which is a key diagnostic feature of cystic fibrosis. From a molecular point of view, the F508del mutation impacts a part of the CFTR protein called the nucleotide binding domain 1. This region is crucial for the protein's folding and stability. Because of the deletion, the protein doesn't fold correctly, which prevents it from reaching the cell surface or functioning properly even if it does. Understanding this mutation has been vital for developing targeted treatments called CFTR modulators. These drugs aim to help the defective protein fold better, reach the cell surface, and work more effectively. Such therapies representing personalized medicine approaches that can significantly improve quality of life for many people with cystic fibrosis who carry the F508del mutation. In summary, the F508del CFTR gene mutation involves the loss of phenylalanine at position 508, leading to misfolded and malfunctioning CFTR proteins. This causes the characteristic symptoms of cystic fibrosis, including thick mucus buildup, lung infections, and digestive issues. Because it is so common, ongoing research continues to focus on ways to correct or bypass this genetic defect, offering hope for better treatments in the future. Always remember, if you or someone you know is affected, consulting healthcare professionals is essential for personalized care and management.